This is the third lesson of Chapter 6, where we're going to be talking about enzymes. Enzymes are biological catalysts that speed up chemical reactions. Now, this particular slide is an embedded video, and I'm not going to play it now because it will not allow you to have sound through the video. I learned that from the previous presentation where I had a different embedded video of the archer. So what I would suggest is you download the PowerPoint and you watch the video there. It's only a couple minutes long, but I'm actually going to go through a very similar diagram with you and discuss enzymes that way. Alrighty, so let's go to that. Now we've seen this graph before. This is the free energy graph showing the progress of a reaction, if you read this from left to right, and on the y-axis is free energy. We have the reactants, which have a higher free energy than the products. So this is a exergonic spontaneous reaction. Before, when we saw this graph, we were focusing on delta G and just kind of understanding what this graph depicts. I mentioned that there was additional information about enzymes, and so now we're going to look at that. The reason that we have two graphs here is to show the reaction, the change in free energy of a reaction with and without an enzyme. To start, the reactants and the products have the same free energy, so delta G is unaffected by the addition of an enzyme. What's different, though, is this peak here. After it kind of goes up this hill and it reaches like a maximum point, that is the energy of activation, or E sub A. This is the energy that needs to be invested in order for that reaction to proceed from reactants to products. It's like if you were starting a business, you sometimes have to make an initial investment to get that business up and running, and hopefully you'll eventually make a profit after you've paid off those investments. So this is the energy investment phase. With an enzyme, the reason that the reaction can proceed more quickly is because the energy activation energy is lowered. I just realized that says energy activation. It should say activation energy. This is what happens when I talk and type at the same time. Little dyslexia. Activation energy. Although I have also seen it as energy of activation. So that would be perhaps also useful to write that down. Just in case you're consulting different resources, it's good to know all the different terms that they might use. Oh no, won't fit. Oh, the struggles. Okay, let's see. I'm going to leave it alone. Okay, so that is the basic idea of enzymes in terms of what they do, how they function. But what is an enzyme? Do you recall learning about the different types of large biological molecules, such as carbohydrates, lipids, proteins, nucleic acids? I hope that rings a bell for you. It sounds familiar. Well, enzymes fall into one of those groups. Ponder that for a moment. Enzymes are proteins. And perhaps this illustration, maybe that triggered that thought for you, because if you recall, proteins can have very complex structures. This would be a protein that has probably tertiary, if not quaternary structure. It's not really telling me what enzyme it is, it's just generic. But when you have all of those additional interactions between the amino acids, like you have the hydrogen bonding forming the alpha helices and the beta pleated sheets, and then you have the interactions between different charges of the R groups, like ionic bonding, and perhaps even multiple polypeptides. The unique structure of the enzyme comes from those interactions. And enzymes typically have one substrate that they can bind to and facilitate a reaction for. That substrate binds to a specific region on the enzyme called the active site. And it's a chemical bonding that occurs there. So they have to be compatible in their structures to bind to the active site. When that binding occurs, the entire thing now is called the enzyme substrate complex. Now, there's a couple theories out there about how enzymes and substrates fit together in their reactions and then facilitate that chemical reaction. The first theory that you've probably heard of is called the lock and key theory. And this is the idea that there's a specific key that fits in a specific lock. And if the key is, different shape or it gets damaged and the prongs don't fit in the right way, then it's not going to open the lock. So similar to that would be what the lock and key theory states, where there's each enzyme has a specific substrate. And let me make a bigger text box here. Not only that, so it has a specific substrate that binds to the active site. And when that binding occurs, there's no shape change of the enzyme. So I'm writing all these notes here. Since they're not on the slide, there's no change in shape of the enzyme during when okay. Now, I'm not sure if you can tell here, but that doesn't seem to be the case in this scenario shown. Doesn't it look like the enzyme kind of clamps down on the substrate after it binds? Well, yes, it should. And in fact, the lock and key theory that was first recognized in the scientific community is now replaced with a different theory, and that is the induced fit model. So with the induced fit model, similar to the lock and key theory, there is still a specific enzyme and substrate that are compatible. But unlike the lock and key theory, it's now known that the enzyme does change shape slightly after the substrate binds to the active site. And it's like it's giving it like a little hug, clamping down just a little bit before releasing the products of that reaction. Okay, so do be sure that you know both of the theories for how enzymes and substrates bind, and now let's look at some examples of enzymes, because this is a generic model here. One enzyme that is relevant to biology and to us is catalase. Catalase works to break down hydrogen peroxide, which is actually a waste product that is produced in a eukaryotic cell. And so we don't want that building up in the body. It can be toxic. So catalase breaks it down into oxygen, O2, and water, H2O. So just kind of a fun fact on the side, since I mentioned that hydrogen peroxide is toxic to cells, and however, we might be using it to clean wounds when we get an injury. I know I grew up doing that. A little bit painful when mom got the hydrogen peroxide out of the medicine cabinet and poured it on an open wound. You see the bubbles and then you feel the burn. Well, the reason that you should not use hydrogen peroxide to clean wounds is because not only does it kill potential bacteria and viruses and things that might be making the wound likely to get an infection, but it can also damage the healthy living cells. So this is a chemical burn somebody got on their hand from a higher concentration of hydrogen peroxide than you might get at the drugstore. It's a little bit more diluted there. So anyways, instead of using hydrogen peroxide, it might be better to use rubbing alcohol to clean your wounds. Now back to in our bodies, catalase is readily available in our bodies, and it works very, very quickly. And this is another property of enzymes. Enzymes are reusable. One catalase can take on many, many hydrogen peroxides in its useful lifetime. And in our body, I'm not sure where I found this fun fact here. Another fun fact, our body can break down about 40 million hydrogen peroxides every second between all the catalases that we have in our body. So that is an incredibly fast rate and pretty amazing what's going on inside of our bodies to keep us alive and protected. All right, so the next few slides just kind of go back to what I was talking about before, defining some of the terms here for you: substrate, enzyme, active site, substrate complex. So I'm just going to slide through this one. And here's another scenario showing how, well, this is kind of good. It shows how there could be more than one substrate that binds to the active site, and that enzyme converts those substrates to the products, changing around the arrangement of the atoms, releasing two different products than originally came into the active site of the enzyme. So sometimes the reaction might involve one molecule as the reactant and might release two products, or vice versa, two reactants, one product, or here, two different reactants, two different products. So there's a diversity of enzymes, and just understand basically how they work, and we'll look at some more examples.